Hello students. In today's session, let us learn about the different types of DNA. As you already know, DNA is the hereditary material in humans and almost most of the organisms except for few viruses. Most DNA is located in the nucleus, which is also referred to as nuclear DNA. Also, a small amount of DNA is seen in mitochondria, which we refer as mitochondrial DNA. So the information in DNA is stored as a code made up of four chemical bases or the nucleobases, namely ATGC, that is adenine, guanine, cytosine, and thymine. Human DNA consists of about 3 billion bases, and more than 99% of those bases are the same in almost all the people. The order or sequence of these bases determines the information available for building and maintaining an organism, similar to the way in which letters of the alphabet appear in a certain order to form words and sentences. DNA base pair up with each other. That is, adenine pairs with thymine and cytosine pairs with guanine to form units called as base pairs. Each base is also attached to a sugar molecule and a phosphate molecule. So together, base, sugar, and phosphate forms a nucleotide. And these nucleotides are arranged in two long strands that form a spiral called as the double helix. The structure of the double helix is somewhat like a ladder, with the base pairs forming the ladder's rungs or the steps, and the sugar and phosphate molecule forming the vertical side pieces of the ladder. An important property of DNA is that it can replicate, that is, it can make copies of itself, and each strand of DNA in the double helix can serve as a pattern for duplicating the sequence of bases. This is critical when cells divide because each new cell needs to have an exact copy of the DNA that is present in the old cell. Moving on to the types of DNA, three major forms of DNA are double-stranded and connected by interactions between the complementary base pairs. These are B form, A form, and the Z form. This image summarizes the different types of DNA. Now, the most common form present in most DNA at neutral pH and physiological salt concentration is the B form. Whatever the Watson and Crick model that we have studied already is the B form of DNA. So it is a classic right-handed or the right side coiling, right-handed coiled double helical structure. A thicker right-handed duplex with a shorter distance between the base pairs has been described for RNA-DNA duplexes and RNA-RNA duplexes. So this is what is referred to as A form nucleic acid. And again, the third form of duplex DNA is called as the zDNA, which is strikingly different. Here it is left coiled. So it's a left-handed helical structure. It is formed by the stretches of alternating purines and pyrimidines, that is GC, 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 especially in negatively supercoiled DNA. A small amount of DNA in a cell exists in Z form. So A form and B form are right-handed duplexes, where Z form is left-handed. Now let us learn about each of them in detail. First one is the most common form, that is the B form DNA. This is the Watson-Crick form of the double helix that most people are already familiar with. They proposed two strands of DNA, each in a right-hand helix, wound around the same axis. The two strands are held together by hydrogen bonding between the bases. The two strands of the duplex are anti-parallel and plectronomically coiled. So the DNA strands, they don't run like parallel, they are anti-parallel. The nucleotides arrayed in a 5' to 3' orientation on one strand align with the complementary nucleotides in the 3' to 5' orientation of the opposite strand. Bases fit in the double helical model if pyrimidine on one strand is always paired with purine on the other. From Chargaff's rule, the two strands will pair A with the T and G with C. As you know, between the nucleotides, it's always the hydrogen bond that exists. So A double bond T, there are 2 hydrogen bonds between adenine and thymine and 3 hydrogen bonds between guanine and cytosine. So it is G triple bond C. This pairs a keto base with an amino base, a purine, and with a pyrimidine. The 2 hydrogen bonds can form between A and T, and 3 can form between G and C. So here, very clearly, you can see the adenine to thymine base pair complementarity. This is the structure of adenine, which is a purine, and this is thymine, which is a pyrimidine. And you can see 2 hydrogen bonds that are formed. The dotted line indicates the hydrogen bond. Whereas here, between the GC base pair complementarity, guanine is a purine and cytosine is a pyrimidine. Here you can see 3 hydrogen bonds. These are the complementary base pairs. The base pairing scheme immediately suggests a way to replicate and copy the genetic information. So between the base pairs, there's 34 nanometer gap, 3.4 nanometer per turn, and about 10 base pair per turn. So 9 nanometer in diameter, which roughly comes around 20 angstroms. And it's a 34-degree helix pitch with a -6-degree base pair tilt and a 36-degree twist angle. So here, very clearly, 20 angstrom is the diameter between the 2 strands of the DNA. And between every nucleotide base pair, the distance is 3.4 angstrom. Since there are 10 base pairs in a turn, it comes around 34 nanometer. Now, next, let us learn about the A form of DNA. A form is fairly similar to B DNA because it is right-handed double helix structure with, again, major and minor grooves. However, as shown in the comparison table below, there's a slight increase in the number of base pairs per turn. So, small rise per base pair, around 20 to 25% shorter than B DNA. The major groove of A DNA is deep and narrow, while the minor groove is wide and shallow. So therefore, A DNA is quite broader and apparently more compressed along its axis than the B DNA. So this marks the difference between B DNA and A DNA. The major difference between A form and B form nucleic acid is in the conformation of the deoxyribose sugaring. It is in the C2 endo conformation for B form, whereas it is in the C3 endo conformation in A form. Now, the next major difference between A form and B form nucleic acid is the placement of base pairs within the duplex. In B form, the base pairs are almost centered over the helical axis, but in A form, they're displaced a little away from the central axis and it is mostly closer to the major groove. The result is a ribbon-like helix with a more open cylindrical core in A form. It is again a right-handed helix, and instead of 10 base pair per turn, we have 1 extra. So it is 11 base pairs per turn and 0.26 nanometer axial rise, 28-degree helix pitch, 20-degree base pair tilt, 33-degree twist angle, and 2.3 nanometer helix diameter. Now moving on to the last form of DNA, that is the Z form of DNA. Z-DNA is a radically different duplex structure with the 2 strands coiling in left-handed helices and a pronounced zigzag pattern in the phosphodiester backbone. Z-DNA can form when the DNA is in alternating purine pyrimidine sequence such as GC. And indeed, the G and C nucleotides are in different conformation leading to a zigzag pattern. Here you can very clearly see the zigzag pattern. The big difference is at the G nucleotide. It has the sugar in the C3 end of conformation like A form, and the guanine base is in syn conformation. This places the guanine back over the sugaring in contrast to the usual anti-conformation that is seen in the A and B form of nucleic acid. We have to also note that having the base in anti-conformation places it in the position where it can readily form hydrogen bonds with the complementary base on the opposite strand. The duplex in zDNA has to accommodate the distortion of this G nucleotide in the syn conformation. The cytosine in the adjacent nucleotide of zDNA is in the normal C2 endo-anti-conformation. This was discovered by Rich, Nordheim, and Wang in 1984. It has anti-parallel strands, very similar to bDNA. It is long and thin as compared to bDNA, and it has 12 base pair per turn, and it has a 0.4 nanometer axial rise, 45-degree helix pitch, 7-degree base pair tilt, minus 30-degree twist angle, and 1.8 nanometer helix diameter. Even the classic bDNA is not completely uniform in its structure. X-ray diffraction analysis of crystals of duplex oligonucleotides shows that a given sequence will adopt a distinctive structure. These variations in bDNA may differ in the propeller twist to optimize base stacking, or in the three ways that two successive base pairs can move relative to each other, can twist, roll, or even slide. Here is a table which compares B form, A form, and Z form. So when it comes to helix, B form and A form are right-handed and Z form is left-handed. When it comes to the base pair per turn, B form has 10, A form has 11, and Z form has 12 bases per turn. Now, apart from A form, B form, and Z form, there are other rare forms of DNA. Let us try to learn about them in brief. First one is the cDNA. The cDNA is formed at 66% relative humidity and in the presence of lithium and magnesium ions. Again, this is right-handed with an axial rise of 3.32 angstrom per base pair. So there are 33 base pairs per turn, and the helical pitch is roughly around 30.97 angstrom. And the base pair rotation is 38.58 degree, and it has a diameter of 19 angstrom. So therefore, this is smaller than that of aDNA and bDNA, because there it is around 20 angstrom. And the tilt of base is 7.8 degree. Here in the image, you can see the cDNA. It is much smaller than A and B DNA. Now, next, let us understand about the ddNA. It is a rare variant with only 8 base pairs per helical turn. These forms of DNA is found in some DNA molecules, which is actually devoid of guanine. So the axial rise is around 3.03 angstrom per base pairs, and the tilt is 16.7 degree from the axis of the helix. Next, we have the eDNA. This is also known as the extended DNA or the eccentric DNA. So eDNA has a long helical axis rise and base perpendicular to the helical axis. Now, deep major groove and the shallow minor groove. eDNA is allowed to crystallize for a period longer. The methylated sequence forms stranded aDNA. Standard aDNA. Excuse me. eDNA is the intermediate in the crystallographic pathway from bDNA to aDNA. Here you can see the eccentric DNA, how different it is from the regular bDNA. To quickly summarize, DNA is an organic compound which has a very unique molecular structure. It is found in all prokaryotic cells as well as eukaryotic cells. And the major different types of DNA include aDNA, bDNA, and zDNA. aDNA, it is a right-handed double helix structure, which is very much similar to B form, but the dehydrated DNA takes an A form that protects the DNA during extreme conditions such as desiccation. Protein binding also removes the solvent from DNA and the DNA takes an A form. bDNA, this is the most common DNA conformation and is a right-handed helix. The majority of DNA has a B-type conformation under normal physiological conditions. zDNA is a left-handed DNA where the double helix winds to the left in a zigzag pattern. It was discovered by Andrews, Wang, and Alexander Rich. It is found ahead of the start site of a gene and hence is believed to play some role in the gene regulation mechanism. Thank you.